the culture of E. coli. E. coli cells, if you look at them under the microscope, you see this like really crazy movement going on all the time. Each of these cells is swimming at 10 to 20 times. Am I supposed to do something there?
Maybe later.
Do it later, sounds good. See you later.
I don't actually see it on my screen at all. So I'm not sure.
It's probably on an extended
I mean,
screen.
on the Zoom, that doesn't show up.
Yeah.
Okay, then I think I'll just continue.
No, you see your extended screen. Just move your cursor.
It's not
Later,
this computer.
I don't know what's
Ah,
the.
there is your mouse.
Yeah.
You could see it
Or just go tab and then
somewhere
hit
into the
enter.
bacteria.
Okay, I'm getting there.
So Navish, just use the tab and then hit enter.
Tab does not seem to work.
Can you just press enter? I guess I will start your video.
Oh, okay.
Oh.
She's really magic.
It's a different screen.
Learned all the tricks.
It's amazing.
Okay, so bacterial cells are swimming at 10 to 20 times their body length per second. And this is sort of, to imagine on the human scale, this is the equivalent of swimming at 60 miles per hour or something like that. So they're doing this amazing feat. So how are they achieving this? So many bacteria like E. coli swim by rotating helical filaments. So in this video, this classic video, the filaments have been labeled by fluorescent dye. And what you see is that each cell has 4 to 5 filaments, and they bundle together. When the cell is swimming, they come together, form a coherent helix, and this helix rotates through the fluid, generating the thrust that pushes the cell forward. So this is how E. coli cells are swimming. And then, of course, the question is, what is driving the rotation of this helix? And that's really what one of the things that I'm really interested in. So if you look at the base of one of these filaments, you would find this complex macromolecular machine that drives the rotation. So here is a cartoon, a much simplified picture of this machine. So out here is the filament, here is the hook that connects the filament and the basal body of the machine, and here is the machine. And if you ever think about electric motors, this machine has all of the corresponding parts that an electric motor has. It has a drive shaft here in purple, it has a rotor in yellow, labeled here in yellow, and it has these stator complexes, labeled as stator units, that remain fixed in the cell, and they generate the force that is driving the rotation. So they remain stationary in the cell. And what's interesting is that E. coli cell envelope has 3 layers. So here's the outer membrane, peptidoglycan, and the inner membrane. And these stator units, they hold on to the peptidoglycan on one end, and they apply this sort of kicking motion on the other end to drive the rotation. And if there's any questions that come along with, please feel free to stop me and ask me those questions. And what's really interesting is that this sort of counter to its name, these stator units, the stator proteins aren't entirely stationary. So if you look at a cross section, so if I make a cross section here, and if I look at the top view, then I have this rotor. This ring is representing the rotor. And what you see is that many stator units work together to drive the rotation. So effectively, the net rotation of this motor is proportional to the number of units that are driving it. And that these units exchange, these stators exchange with a pool of free units that are floating around in the inner membrane of the cell. So there is a constant turnover going on where free units can take their place and the bound units can leave. So if they're, say, damaged or something happens, so there is a self correction mechanism in there. So with this system, Actually, here is a cryo-EM structure of just the basal body of this machine, and you can see that it's just like really beautiful and complex structure, which remains a source of mystery and inspiration for my lab. So one of the questions that we are really interested in is how does this motor that is driving these flagella of bacterial cells, how does it cope with sudden and dramatic changes in load? To give you one example, imagine the cell is swimming around in water and suddenly it enters a very highly viscous environment, which means that the load on the motor would go up. And what does the motor do in that situation? Does it stop spinning? What happens? And so this work I did mostly as a postdoc in Howard Burke's lab, and this led to wonderful collaboration with Rob Phillips and Yuhai Tu, who has magically showed up here. And it's being continued in my lab by Shabdali Sawa. So I want to take an analogy to think about this problem. So imagine you are in a car, a beautiful red car, and you're driving down the highway, and it's a perfectly flat road. And you so kind of know from experience that your engine has to supply a relatively small amount of torque to keep the car going. But then suddenly, if you have to go uphill, most of the cars in the US at least have an automatic gear shift, which will adjust the output of the engine with the demand. So the question then we are interested in, can you do the same kind of thing with bacteria? And the answer is yes, you can. So here's an experiment that a previous postdoc in the Berg lab did. So he took a bacterial cell and he removed all of its filaments, leaving behind a very short stub shown here. And he attached this cell on a surface. And when you do that, you can imagine that this short stub, so there's a motor at the base of the stub, it was driving the rotation of a whole filament, but now the filament is gone, so it's working at a very low load. And then what he did, he took a plastic bead in an optical trap and suddenly attached that bead to this little stub. So what happened is that between the situation on the left and on the right, you have suddenly increased the load on this motor quite dramatically. And when you look at the response of the motor, you see something really interesting. So here's time on the x-axis, and the speed of the motor is on the y-axis, measured by the rotation of this bead. And this arrow represents when the stimulus was applied, when the bead was first attached. And what you see is that initially, this bead rotated at a very low speed, at about 5 hertz. But over time, in just a matter of like 2 or 3 minutes, the speed increased, ending up at about 50 hertz. So it's like in your car, initially your engine is kind of struggling to keep you going, but then it speeds up. Yeah, Noah.
On this plot, are the vertical stripes switching from 50 to minus 50?
Yes. So what it's this motor is bidirectional, so it switches to between clockwise and counterclockwise. So this is just representing the clockwise rotation.
Yeah, so if you actually put dots there wouldn't be a lot of dots in between. It would be sort of two curves. Yeah.
Yeah.
So the time scale of the switch is much longer than the period of the rotation, right?
So this motor is rotating, as you can see, at about between 10 and 50 hertz, and the switch happens basically instantaneously and switches back pretty fast, but about a second or so.
It stays in one correlative for more than a second, right?
Yeah.
So I'm very confused. You've added the sphere, which makes it much more difficult to drive. It's got much more viscosity.
Yeah.
And the response of the cell is to increase its frequency, so it's working not only against the additional viscosity, but at a much higher rate.
Yeah.
So it's not the same as a car where you keep your power output constant. Here, it's dramatically increasing its power output.
That's right.
So the analogy doesn't work.
So analogy isn't perfect.
No, but
It fails completely.
No, but it's related to the question, what was the rotating speed before you had the beads?
So before you add the bead, it would be rotating at about 300 hertz.
No, so that's,
Yeah.
I see. It came down.
Yeah.
I thought it was starting at 5 hertz.
Oh, I see. No, so
I see.
before you add the bead, which we cannot see, it's spinning at about 300 hertz
I see.
under negligible load.
Okay.
You add the bead, it goes down to 5 hertz.
Thank you very much.
Yeah. Any other questions? Okay, so we want to know what is the physical and molecular mechanism that is underlying this gear shift analogy. Okay, so this is when I came into the project and I asked, okay, how can we change the load on the motor in a way? So we thought of like an ideal experiment. What would be an ideal experiment? So if you think about load on the y-axis versus time on the x-axis, we would like to be able to change the load instantaneously. So something like a step function, but we would like this change to be reversible, and we would like to be able to control the amount of the stimulus so that we can study the response to varying degrees of stimuli. And this is a challenging ask, but we were able to come up with an approach that satisfied these criteria, and this approach is electrorotation. And so let me describe how that works. So what we are doing is that we flip the previous experiment upside down. Instead of attaching the cell to the surface, we attach the stub to the surface. And the motor is still working, so instead of driving the stub, the motor is now driving the whole cell. And you can imagine that it's a tiny motor driving this big cell. It's a very large load that it's starting with, and it rotates at a very low speed. So the motor has to supply a large torque. And what we then do is to apply an external, very high frequency rotating electric field on this cell, which applies an external torque, we call electrorotation torque, in the same direction as the motor torque. So essentially the motor is trying to spin the cell, say, one direction, it's working hard, but then you're providing it assistance. So by providing this assistance, we lower the load experienced by this motor, and then the motor torque goes down and the cell spins at a high speed. So essentially, just by turning this external field off or on, we can change the load on the motor quite dramatically. Yeah?
Why is the cell responsive to a rotating electric field?
So this is related to electrophoresis. So what's happening, I don't know the full physics of this, but what's happening approximately is that the external field is causing transient polarization in the cell, so it's inducing a dipole, but we're rotating the field at megahertz frequency, and the induced dipole lags behind the external field, and the interaction between the external field and the induced dipole causes a torque.
Then
Yeah.
how do you know that you're not acting a torque on the motor? Or like, so you're applying this electrorotation to the cell.
Mm-hmm.
Does it have any effect on the motor?
So it has an effect on the motor in the sense that it's lowering the load.
Yeah, but I meant a more direct effect. Like you mentioned that you're inducing a dipole, so that it makes sense that maybe you could make a length scale argument, like a separation of scales of.
Yeah, so what we believe is happening is that this, because we're spinning it at megahertz frequency, 2.5 megahertz, this is so much faster than anything happening or sort of relevant timescales inside the cell, that on relevant timescales, your average field is essentially zero, because you average a circular field, it's basically zero.
Okay.
Yeah, so that's what we believe, and we can sort of see what happens to the cell, and the cells are completely fine.
Mm-hmm.
So we believe that they effectively just experience a torque and nothing else. Yeah.
So you do this, you don't apply a uniform, the field is in the direction, is it tangential, or which direction is the field?
The field is spinning at megahertz frequency.
Right, but if you put the arrow, the vector for the field on there, which way would the field point?
So the field would be like this.
Oh, so it's along,
But
I
then
see, so the
spinning-
dipole, got it, okay.
Spinning all the time.
Yeah.
Yeah.
Okay.
Much faster than what the cell can.
But then the induced, if the field's along that direction, how is there a torque on, I mean, is the induced dipole along that direction too?
So the field is in this direction, but it's spinning at megahertz rates.
Uh-huh.
So it
Okay, so it induces a dipole,
right,
I
which
see.
is also spinning at megahertz rates, but it's
but it lags because,
It lags
okay, it lags,
behind.
I see, okay,
Yeah.
okay.
okay. So this is what it looks like. So this is instantaneous, reversible, and controllable. This is what it looks like in action. So when the field is off, the cell spins at a low speed, and every time the field goes on, the cell spins at a very high speed and the load goes down. And we can keep playing this game over and over however we like. And what's important here to notice is that while the field is on, we cannot observe the state of the motor because it's being driven by the external field. So we can only observe it during the off period, so to speak. So this is an example of one experiment. So again, time is on the x-axis and speed is on the y-axis. And every experiment consists of 3 phases. You have a initial phase, we're just observing the initial state of the motor. We turn the field on, reducing the load, and then we turn the field off, increasing the load. And this is 6 minutes and then 6 minutes. And during this on period, because we cannot observe the state of the motor, we turn it off occasionally. So every 8 seconds, we turn it off for 1 second to see what the motor is doing on its own. So initially, this cell and this motor rotated at about 10 hertz, 11 hertz. And then we lower the load using electrorotation. And when we observe it briefly, we see a stepwise reduction in the rotation speed of the cell. And the interpretation of these data is that each of these steps corresponds to the release of one of these stator units that are driving it. So remember, I showed you that at any time, basically you can have like 10 or so of these stators that are driving the motor. And so effectively, when you reduce the load, the motor is letting go of these torque generating stator complexes, effectively to match its output with the demand. And then at the end of this period, we can turn the field on again, which increases the load. And now we can really see a stepwise increase in the rotation speed corresponding to a sequential addition of stator complexes. So what I find really interesting about this experiment is that this is not a molecular, single molecule experiment. We are working with whole cells, but we are getting a single molecule resolution. We can watch the binding and unbinding of individual proteins. Yeah.
So do you interpret this as 4 or 5 stators then? Like
Yes.
Incrementally. So
Yes.
you roughly go down by a factor of 2, from 10 to 5-ish?
So let me actually show those. So we can fit steps on these data. We can extract the number of units. So what's happening in this case is that we start with about 10 units. We went down to 0, and we went back up to 9. So that's what's happening in this case. So essentially, the message of this first part of my talk is summarized here, that the motor adapts to changes in load by remodeling its stator itself. So the stator itself acts as sort of an automatic gear shift. It's like you're driving down, but instead of changing gears, your car is adding or removing pistons, which does actually exist. Some manufacturers do make something like that in which they can couple and uncouple pistons. So that's sort of the mechanism that we are seeing here. Yeah.
So in the initial setup, when there is a bead attached to the motor, you even showed in your plots that it switches direction very quickly.
Yeah.
Does not happen in this opposite case when the motor is trapped.
No, it does. We are working with a strain that is logged in one rotation. So we mutated its gene that's responsible for switching. So it's always going in one direction. Yeah. Okay.
But then it should saturate, right? Because you cannot add as many stators as you want.
That's right.
So here you were decreasing load, but in
Yeah.
the previous experiment you were increasing load dramatically.
That's right.
It was still able to adapt.
Yeah.
So can you probe that with this new system? The fact that you saturate the engine in terms of adapting to more load.
We can. So essentially what we can do, so in this case, we start with saturated system, about 10, because we start with high load. So we could manipulate it to start with an intermediate load and then go either way by increasing or decreasing the load. Yeah, so we can do that.
Can you also not drive it against its
Yes.
preferred chirality?
We can, yeah. But in this experiment, we wouldn't see any change because it's already saturated. Yeah. Mm-hmm.
So the switch of chirality, it is the switch of all of the stators, or there's some mechanism on the shaft that is switches, the stators are still doing the same thing?
Yeah, that's a great question. So stators are always doing the same thing. By the way, we understood this only like last year or 2 years ago from a structure, that paper that was published. Stators are always doing the same thing, and it's the rotor that's going into this really dramatic conformation change to allow it to spin in one direction versus another when the switch takes place. Yeah, that's a great question. Okay, how much time do I have?
Well, we started a little bit late. Maybe we can eat into the coffee break a little bit, if that's okay.
Sure.
Okay,
At
so
4-ish. Okay, maybe another 10 minutes.
okay, so I will skip a bunch of slides. So, but what the message of these slides is that we not only understand this process qualitatively, but we understand this quantitatively. We have models that work really well with the data, and I will talk about this slide. So what does this mean for the molecular mechanism for why this is happening? What we think is happening is that at low torque, when the motor is working against not so much resistance, these units are sort of in a relaxed state. But if the torque certainly increases, then they are physically stretched in a way that makes it harder for them to unbind. So that they stay on longer, and overall the number of bound units increases over time because they're always dynamically coming in and out. So if they stay bound longer, you're essentially increasing the lifetime of each unit. So that's our sort of simple picture of how it's working on the molecular level, and my graduate student is working on figuring out the mechanism sort of on a more protein level. Okay, yeah.
And just also, so the time scale over which it returns to its, natural saturated state, that's really this molecular time scale of binding and unbinding, or where is that coming from?
So the
You
time
showed
scale over
steps.
which it increases.
Yeah, you showed
Yeah.
each step taking some amount of time.
Yeah, so that's the stuff that I kind of skipped over. But essentially the on process is independent of load because they are just coming on. But the off process is sensitive to load. So when they leave, that rate is determined by the external load. And so we can measure those things, and it fits really, really nicely.
Navish, so the coming and binding to the peptidoglycan is basically a stochastic process, right?
Yeah.
So is it like the cell start producing more stator proteins and stuff when you do this? So is it like purely how should I say this? Is it an active process, or is this a passive process where it's like by chance I'm collecting more molecules and storing them because they don't leave?
Yeah, it's the latter.
Oh,
So what you
there's
just
no
said.
sensation that now my in the cytoplasm, say, I have less molecules, now I have to switch on and produce more.
So this is happening on timescales that are way faster than any of the gene regulation, any of the real biological regulation to take place. So we think that this is a purely biophysical mechanism that is built into the structure and function of this protein. Yeah.
Okay.
Yeah.
So more just zooming out, and this is maybe a very naive question, but in vivo, these are typically bundled, right?
Right.
Rather than single. So
Yeah.
the base of the motor, the configuration of the bases is static, right?
Mhm.
So that so there's a lot of sliding with respect to other flagella in the bundle.
Mhm.
And so can you maybe just comment on connecting, what effect that would have in the more physiological state where you have multiple
Right.
flagella
Yeah.
that are,
Yeah, so I think what happens in a real swimming cell is that the bundles interact with each other, but if the entire cell's environment changes, then each bundle would experience a higher load, and they would each filament in the bundle would experience a higher load, and they would all need to step up, so to speak, and this process would allow them to do that. And also what happens a lot is that flagella break and they're constantly growing. So if you have a really tiny flagellum which is not producing any torque, then you don't need to be spending energy with these units at the base of this flagellum. So it also modulates for that process. Okay, I want to take just 5 minutes to talk about some new stuff from my lab, which is collective motion of non-modal bacteria. And this is largely driven by Eric Dudebou, who's an undergrad in my lab, and it's a collaboration with David Blair at University of Utah. So bacteria move in a variety of complex environments, and one of the very important of those is moving over surfaces, bacterial spreading over surfaces. And one of the classic mechanisms that has been very well studied, and we actually saw some examples this morning in Olivier's talk, is swarming. So if you take a bunch of, I'm just going to talk about E. coli as an example. If you take a bunch of E. coli and you place them on a soft, take like a small amount, put them in the middle of a soft agar plate, which is substrate with nutrients for them, they will spread out. And they spread out, and this is pretty well understood, they spread out in this collective motion, which is called swarming. And if you say this is an example of a zoom in, at the edge of the colony, you have like gazillions of cells, and each one of those cells have their flagella. They're constantly driving their flagella to move forward. And this is a video taken from Howard Berg's lab, which shows at the edge of the swarm, these cells are in constant motion. So this is actually has been basis of a lot of work in active matter. But then I met with David Blair last year, and he showed me these data in which you have cells that do not have flagella. So let me remind you what I'm talking about. So here's the structure of the filament. So this is the filament, and here is a protein called flug K, which connects the hook with the filament. So if you don't have flug K, you don't have the filament. Similarly, here is the the stator proteins that I talked about, mod A, mod B. So if you don't have these, you have the filament, but it's paralyzed. It doesn't spin. Okay, so those are the two examples that we're going to think about. So Dave showed me these data in which he did experiments. So these are wild type cells, and if he plated some wild type cells and they swarm out. So this is classic swarming behavior. Yes.
Staphylococcus?
This is salmonella.
Salmonella.
Yeah. And then he had cells with this delta flug K mutation, so they're missing the connector, so they have no filament, but they still spread out. And not insignificant as compared to the wild type, it's actually quite comparable. And then I went back to my lab and we thought, okay, can we do this in E. coli? And turns out that yes, you can do this in E. coli. And in fact, even more. So this is wild type. These are classic swarming cells, and you can zoom into them. You find swarming cells. Here is delta flug K, non-modal cells lacking flagella, lacking filament. And here is delta mod A cells, which have flagella, but they're paralyzed. They don't spin their flagella. In all of these cases, these cells are spreading out. And in fact, one could say that this delta flug K are spreading out faster than the wild type swarming cells.
How long did it take them to get that far?
So this image is probably about 12 hours after inoculation. So I have a really nice video that Eric made, which shows, so initially you inoculate, then it takes some time for them to grow to a certain density, and then they sort of start spreading out in these like really beautiful patterns. And so this is what we are trying to understand in my lab. And so what we think is happening is
They are dividing, right?
they are dividing.
So
Yeah, so there is growth, but this is like, if you look at sort of
What's the time?
over here, so you don't see a lot of cells, you still see a front spreading out.
Right. So Navish, do they produce a lot of surfactant? And sometimes they do produce surfactant and glide over that, right?
Yeah, so we think it's something like that happening. So let me so it's actually surfactant is bad. So let me show you just very preliminary data for a few physical factors that we have looked into. So surface wetness really enhances colony expansion. So here we're changing the percentage of agar that we're using. So if you use less percent of agar, your base is more wet. So they expand faster. If you use more agar, then you increase the stiffness of the surface. It's less wet. They expand slower. Surfactant actually inhibits the expansion. So here we are
They
using.
can produce surfactant, right?
So E. coli cells are not known to produce surfactant for this stuff. So here we're using tween as a surfactant. If you use 10 to the minus 4 molar, you see we've significantly suppressed their expansion. But if you reduce this level of surfactant, they start spreading more, and they spread most when you have no surfactant. And one of the things, this is already that Dave had looked into, it's really dependent on the sugar that you use. And so we believe that the fermentation of these sugars is something that's really aiding this process. So if you use glucose, which is a sugar source of choice for bacteria, which can be fermented into lots of different byproducts, they expand really fast. If you use sucrose, which is not fermentable, then they don't expand. If you use arabinose, which is somewhere in between, then they go sort of halfway. So our working model is that the production of osmolytes by the fermentation of these sugars is creating an osmolarity gradient at the front of the cell, which is pulling up water from below and pushing the front forward. And we can sort of see some features of this process in action. So this is still very preliminary, so I welcome feedback if you have any ideas. So let me just wrap up with the take-home messages. So first part of the story, I talked about how stators act as dynamically remodeled to adapt to external load, and that non-motile bacteria spread over surfaces through fermentation-driven gradients. So this is my lab, and I also want to thank funding sources, and thank you. And I'm happy to take one or two more questions if there's time.
No time. But we do have a break after the next speaker. So I would like to introduce Elizabeth Jerrison, and thank you again so much for the fantastic presentation.
So,
Let's see if we can Zoom connect. Yes, there's the Zoom.
they have it.
Make sure that they are multiplying.
They are dividing, and we are sort of trying to uncouple.
Yes. Use a cell cycle blocker.
So they stay connected, they grow but stay connected.
Are you perhaps?
Oh.
Something is happening on my screen, so.
It might be that it can only be one person.
Yeah.
There we go.